Okay, guys, we're going to start with the organelles, and the first one up is the nucleus. Um, you'll recognize it as the control center of the cell, but more importantly, it controls the production of proteins. So its job is to house all the DNA in the cell and then to tell the cell to make proteins, because proteins carry out many functions within the cell. The membrane on the outside around the nucleus is called the nuclear membrane. Inside the nucleus, you're going to find your chromatin, which are small strands of DNA that houses your genes and your chromosomes. Your chromosomes give you your character traits, your hair color, your eye color, and those traits are inherited from your parents. The nucleolus is the organelle inside within the nucleus, and its function is to produce ribosomes. Ribosomes produce proteins and enzymes. It's found inside the cytoplasm. Those are called free ribosomes. And then you have your ribosomes that are attached to the endoplasmic reticulum, which is called the rough ER. So the rough ER is an internal membrane where lipids and proteins and other materials are assembled and exported out of the cell. So you have two types of endoplasmic reticulum, your rough ER and your smooth ER. Your rough ER is a portion that makes proteins and appear rough looking because the ribosomes are attached to the outer surface of it. It's usually housed very close to the nucleus. The smooth ER does not have ribosomes on it, so that's the difference. One difference between the two. The other thing is that smooth ER function is to detox the body. So if you drink alcohol or take drugs or have taken something that's going to be inside of your body, the smooth ER processes it and tries to take it out of the cell, and that's called detox. So its purpose is to detox the cells. The Golgi apparatus is referred to as the post office of the cell. It sorts and packages and ships proteins out of the ER. So it's referred to as the post office of the cell. It's folded up, looks like little flat pancakes. Your lysosomes are your janitors. They clean up accidents, so to speak, in the cell. They break down lipids and carbohydrates and proteins, and any type of cell that has maybe dysfunctioned, and the lysosomes are full of enzymes, so those organelles will, this organelle helps clean up the rest of the cell, hence being the janitor. Your vacuole is found in plants and animal cell, but they're extremely large in an animal cell. They store things like water and salts and proteins, and it's a sac-like structure, a lot bigger in the plants than in the animal cell. Um, the water within the vacuole inside the plant cell has pressure, which gives the leaves and flowers support, things that are heavier for the plant to hold up. The vacuole will push out on the tissues and help give that structure. And in your single cell organisms, such as your bacteria and paramecium and stuff, the contractile vacuole contracts and pumps excessive water out of the cell. And this is what the contractile vacuole looks like. This is a paramecium with the cilia on the outside, and then your contractile vacuole that opens and closes, that causes things, the bacteria and stuff, to move. So it moves the water out in and out of the canals. Your mitochondria is called the powerhouse of the cell. It has a couple of features that we need to look at, and one would be the folds inside of the mitochondria. and the folds are folded up, and increases the surface area so that the more folds that are inside of it, the more ATP energy the mitochondria can produce. So you're going to find the mitochondria in cells like your muscle cells. A cell that would need a lot of energy to perform their job. So another example of that would be a sperm cell. Um, it needs a lot of energy to swim through the fallopian tubes to get to an egg to fertilize it. It's sausage shaped, or oblong shape, and the folds on the inside are called crista. And these things are, the folds are there to increase the surface area of the mitochondria. In a plant cell, you're going to find something called the chloroplast, and the chloroplast is the solar power uh, part of the cell. It collects sunlight so that it can produce energy for the cell to use, and it transforms light energy into chemical energy. Um, and then the mitochondria changes the food molecules into energy. And the chloroplast is located in cells of only plants and some green one cell organisms that we know called algae. Your cytoskeleton is made up of your microtubules and your microfilaments, and they're very fibrous. They stack up. They help give the cell structure. It's like a little folded up matrix. It gives it shape. The cilia and flagella is known for locomotion, and locomotion stands for movement. So the cilia are hair-like, very small, usually found on the outside of paramecium. Was the example earlier. You have cilia in your lungs, and if you're a smoker, then you smoke and you paralyze that cilia, and you're unable to um, keep your lungs clear. You'll have a hard time coughing, and that's why people get something called a smoker's cough. Flagellum is you'll find in animal cells and prokaryotic cells. The flagellum in the animal cells is usually referred to as your sperm, and the sperm has a whip-like tail that helps it use for motion. This is a picture of the cilia. You can see that they're very long and hairy looking, and these are taken from electron microscope, so you get a very sharp image. Okay, we've talked a little bit about the cell membranes, and this coming week, we're going to talk a little bit more about them. But the cell membrane, remember, gives the cell, controls what moves in and out of the cell. So it's referred to as the plasma membrane. So we have plasma membrane, cell membrane, which is the same thing. And its whole function is to promote homeostasis. And remember, homeostasis is to maintain balance. There's a couple of things that you want to make sure that the cell maintains homeostasis, and it's your temperature, the amount of water that's in the cell, and then you want to make sure that your pH within the body is regulated. So, but mostly the homeostasis is dealing with the movement of water and diffusion of things in and out of the cell that we'll talk about next week. I'm going to talk a little bit about the cell wall again, because we did talk about that this week for this test. Cell wall is present in animal cells. So plants, fungi, most bacteria, uh, and other cells have a cell wall, and its main function is to provide protection for the cell. It's located outside the plasma membrane, and it's non-selective. So in the diagram here, and we'll go over a diagram because you have two diagrams on your test. You will have to label a plant cell, and you are going to have to label a animal cell Monday. So remember that your vacuole is the larger organelle in your plant. Your chloroplast has little stacks of chlorophyll here inside of it, your mitochondria. Your smooth ER is going to be near the rough ER, okay, and your rough ER is going to have ribosomes on it. Um, the cytoplasm is your jelly that holds everything in place. Your cell wall gives it support. Your nuclear envelope or membrane is going to be around the nucleus. The nucleus is going to be nucleolus is going to be inside the whole container of the nucleus, and then your Golgi apparatus, your post office of the cell is going to be stacked up, looks like stacks of pancakes, and those package and move things out. Remember the difference. Your bacteria cell has your DNA and your ribosomes. Your DNA is curled up inside of the cytoplasm. Um, no nucleus. So, and then it has a flagellum, and this is the tail, whip-like tail that causes it to move. Some other things that we talked about in class this week, and we looked at pictures of microscopes and or microscope pictures and stuff. Remember that your magnification on your microscopes, you have an optical lens, and it's at 10. And then the lens, the objective lens that you're looking at, you have to look at the number on the objective lens and multiply those two numbers together. So if you have a 4 on your objective lens, then 4 times 10 is 40, and that's your magnification. And then this goes into what we're going to talk about next week. Make sure that you remember um, independent and dependent variables. Independent variable in the scientific method, independent is the one that you're going to change, you're going to manipulate. Is usually formed or shown on the x-axis, and then y is going to be the dependent. So we talked about x causing y is a good way to remember that. X is always something that you're in control of, you can change, and then Y is the outcome of your experiment. The other thing is the control. We have to have a comparison when we do an experiment, so we have to have a group or something that is the same in all of the experiment that we can compare to. In our lab, we looked at with our macromolecules. We tested everything on each group had water. So water would have been our control group to compare our lipids test and our carbohydrates test to. So our water was our control in that group. Um, look back over your notes, look back over your vocab, and I will see you tomorrow.